All right. So we're only going to do a couple of questions on the Kahoot today because we haven't gotten to a lot of chapter 16. So we'll do a couple of chapter 16 questions and then cut it off. And then we're going to jump into finishing our lecture on chapter 16. I just want to get a couple questions in just so that we can get some attendance on the record. 16. Okay, so unless you have taken physiology, you might not have learned anything about the immune system and how it works, especially with the adaptive immune system. It actually gets pretty complicated, and following how the things work and operate with one another can get, well, it's a lot. We'll just say that, okay? These are topics that I spent multiple semesters learning, and you get one chapter on it. And so we're going to cover the basics of it, but I want you to keep in mind that what we're covering might feel like a lot to you. This is nothing. This is nothing compared to what is actually going on. When we actually learn about the immune system and how it functions, we're not just saying it's cytokines that send the signals. We're learning about each cytokine and what each cytokine does, what their names are and how they're different from one another, and what each signal actually does to control this stuff. And I'm trying to leave that stuff out for you guys so we can get the big picture stuff. One reason I'm bringing this up is because when you go out to find videos or something that are going to try to explain how the immune system works, you're pretty much primarily going to be finding videos that explain everything and not just like the watered down basics of it. And it's going to be really hard to kind of parse through that stuff and compare it to what we actually have to learn for our course. So I'm going to try to cover that stuff as much as I can in our version of it, because it just gets really overwhelming when you start looking outside. And and I understand that. I want to let you guys know that I understand that. All right, so let's pull up our Kahoot. We're going to do just 2 questions, and that's going to be it. Nose is itchy today. There. We might not get to the questions today. That's all right. I'm thinking it's crying over that. But because I would rather start on chapter 17 if we have the time. So I might just run through them. But if I feel like we have the time, I'm going to put names up there anyway. All right, we're gonna go ahead and get started with our 2 questions. All right. Our question 1, where do all lymphocytes begin development? All lymphocytes start development in the same place. What is that place? All right, so they all start out in their development in the bone marrow. Remember that all of our lymphocytes are essentially going to be derived from the hematopoietic stem cell precursor that exists in the bone marrow. After it goes through certain stages of development, T cells will move into the thymus to continue their development, only T cells. B cells stay in the bone marrow and finish the development there. But once they have finished that development and they've turned into actual just naive, waiting to be activated cells, then they will move it to the lymph nodes, or T cells will also just circulate in the circulatory system. All right, next question. All right, during maturation, lymphocytes weed out anti-self clones and will only allow appropriate clones to proliferate. There's a term for this. We haven't gotten to it yet in the lecture. Of course, this would be the second question. Yeah, so again, make guesses, base it off of the notes. We will talk about it today in lecture. All right, so this process is called clonal selection. It's basically weeding out all of those anti-self guys. So we've talked about how your cells can basically make all different ability to attack all these different antigens. We say like 8.6 billion or something like that. There's billions of antigens, and that includes the ability to attack self molecules. We have to weed those guys out before we send them out to start searching for antigens. And that is a process that we call clonal deletion. And that basically allows us to develop immune tolerance to self molecules. And we'll talk about that today. And then after we have our whole repertoire of guys that don't attack self, hopefully, that those guys, there's one cell for each possible antigen. When we find the antigen, whether that is, red cap marker, like we were saying, if it's red cap marker and that's the antigen that I recognize, we see red cap marker, and the cells that see red cap marker will be the ones that will be proliferating, not the ones that see blue markers, not the ones that see, yellow cap markers, not the ones that just see black markers or anything like that. Just the ones that would see the red cap marker will proliferate. And so we will only selectively proliferate out the ones that see that one antigen. And then those proliferated cloned out cells will then differentiate into their roles. And we'll talk about what those roles are in the lecture today. But that whole process is called clonal selection. Okay, there's our little answer for that one. And then I'm going to get out of this and we're going to start the lecture. I think I can get this to cooperate with me. There we go. All right, cool. Okay, so we are on chapter 16. Let's try to finish that one up today. We left off last time, we had mentioned MHC. So I want to retouch on that really quickly, and then we will move into the rest of the talk. So MHC basically stands for Major Histocompatibility Complex. Another word for that we use sometimes in science will be human leukocyte antigen, or HLA. I won't test you on HLA on your exams. I will test you on MHC. And so there's 2 different versions of MHC. The MHC that is just the hand that holds out things from outside, or the MHC that is the hand that holds out things from inside. So there's 2 different versions of MHC. If it's held up from by MHC2, that means it came from outside. If it's held by MHC1, that means that it came from inside. And the fact that those MHC are going to only hold antigen from inside or outside will help drive the response to attack things that are inside or outside. Okay, so let's get into how we'll recognize that. So on our cells, basically, you might have a cell that's infected or you might have a cell that's an antigen presenting cell. Let's go with our antigen presenting cell. It found stuff that was hanging out outside. It's a nice globule shape like this, whatever. And it has found something from outside. It eats it up. And then it will put that outside stuff on MHC2. It doesn't care what it is. It doesn't care that it is from COVID or from flu or from a bacterial infection or what. It just knows that it picked it up from outside. And because it came from outside, we are putting it on, holding it out on our MHC2 arm. Okay, so this is our outside stuff. How do our lymphocytes engage with the antigen when it's being presented that way? Well, what happens is our T cell receptor will come in and recognize the antigen. So here our T cell receptor will recognize that. And it is a specific recognition on that end. So MHC can hold up anything, but that T cell receptor specifically recognizes that specific antigen. So this T cell receptor is a T cell receptor that recognizes COVID spike protein, okay? None of the other T cells with receptors that recognize flu spike protein or HIV spike protein or molecules on the surface of tuberculosis or anything like that, those guys can't interact with this. This is a very specific interaction with that receptor. We're recognizing that specific antigen being presented by MHC2. So this is a T cell. And specifically, it's going to be a T helper cell. We'll get into that in a moment, all right? And I'm going to draw out how all of this works once we introduce our basics, okay? All right, so the T cell receptor and how it interacts with that specific antigen. We have a lot of variability at the end of that T cell receptor. This is what's going to be interacting with the shape and the size and the makeup of that antigen. So all of that is happening. So this is our T cell. This is our T cell receptor right here. And this is our antigen binding site. This is where we're going to have all that variability. B cells, they interact with antigen on a receptor on their surface as well. So for B cells, they have a receptor. Tell me that this doesn't look like something that we've already introduced. Antibody, right? And so we introduced antibody during our ELISA stuff in lab, and we drew it to look something like that. And go figure. So their receptors on B cells are just membrane anchored antibody. That's all they are. But again, it's the same exact concept. Antigen binding site at the top, and that's where all that variability is. Okay, so that's how the receptors work on the surface of those cells. Both of them have that variable region at the top and then a constant region at the bottom. So let me show you a picture of what immunoglobulin looks like on our B cell receptor. So immunoglobulin can be the version that we're talking about, that's the receptor, or it can be the version that we are anchored, that we're actually making antibody with. So you can imagine that if you're a B cell and this is what your receptor looks like, and it's anchored on the surface of the cell, that now that we have interacted with antigen, with our antigen binding area on our receptor, that the antibody that we're going to crank out is going to match that pretty much exactly. So we're going to just mimic what our receptor looked like, but create a soluble, version of just these proteins being made as a result. So that whole structure, whether it's membrane bound or secreted as antibody, that's the immunoglobulin. So you guys probably heard about patients that get immunoglobulin infusions. So that's going to be like free, this version, the antibody version of it. Oh, come on. Yeah. So that's usually what we're talking about. The structure of that, whether it's in the receptor version or the secreted version, the structure is the same. All right, so it consists of two major parts. The first part, these parts. This is the heavy chain. All right. Then the little parts that are out on the side, we call these the light chain. We've already talked about how at the top part of these guys, where we're actually going to be binding antigen, That's where all that variability is going to come from. The 8.6 billion antigens that we can interact with happens because of what's happening at the top part there, top of the Y. That region at the top of the Y is called the variable region. Pretty straightforward. And the bottom part is called the constant region. We don't get a lot of variability here. There's not 8.6 different versions of the constant region. There's actually only 6. We're going to talk about what those 6 are and why they're important. But basically, the variable region determines what antigen you're going to bind to. The constant region determines what functions you can have in the body. Okay, so the variable region binds to antigen. The constant region controls the function of that immunoglobulin. If we have an immunoglobulin type D, for example, which we would call IgD, that is the receptor version. We're going to go over all of them in a little bit, but I'm just giving an example. So this is our receptor. Or when we have other ones like IgM or IgG that are just secreted into the bloodstream and float around and just interact with antigens when they see them. And when they bind to them, the antibody has 6 different functions that we're going to get into later as well. But this is just the gist of the shape and how that shape is going to be involved in the function. So we know in B cells, their B cell receptor immunoglobulin can also be secreted as an actual antibody. As it turns out, when we're actually looking at the structure of our antibody, that variable region has another name for it. It's called FAB. You do need to know this. It's in another slide, but I'm just going to bring it up now because we're already looking at the structure. FAB is what determines that it is an antibody attacking a specific antigen. The FAB is the variable region. The constant region has an even easier name, FC. FC, C for constant region. Okay, and so that's how we determine those based on like their structure when we're analyzing their different structures with like crystallography and things like that. The FAB will be our variable region, and the FC fragment, F is for fragment. But the FC fragment is going to be determining the constant region and therefore the function of our immunoglobulin. Okay. The T cell receptors are very similar. They're a receptor that is bound on the surface of the T cell, and they have a weird shape that you don't need to memorize or anything like that. But in the end, it's very similar because we will have a variable region and we will have a constant region. And the variable region is the part that interacts with the antigen, and the constant region is what's going to, for the case of our T cell receptors, they are never secreted. They're always on the surface of the cells. They're only ever surface receptors on cells. Only B cells secrete their receptors as antibody. T cells are important for cell-mediated immunity. In the T cell side of things, the cells are doing the work. On the B cell sides of things, B cells are making antibody. The antibody is doing the work. So the cells aren't actually doing the work of interacting with the pathogens, in the B cells case. But in the T cells, they are. So the T cells, the cells are doing the work, so we call that cell-mediated immunity. In B cells, because the antibody, our soluble protein, is doing the work, we call it humoral immunity because it's in the humors. It's in the fluid part, okay? But T cell receptors, they have a similar concept with that variable and the constant, and that's basically dictated by, the shapes and your genetic makeup and stuff like that. Okay, so with our variable region, how do we get so many different variations from when you only have 20,000 genes all together in your cells? How do we get 8.6 billion different possibilities, or whatever that number is? So we get all these billions of possibilities by reassorting our genes. So all of your variable regions are made up of a combination of V, D, and J segments, roughly speaking. And you don't need to know that. I'm not going to test you on it, but we'll take some V genes and we'll take a D gene and we'll take a J gene. And you might have 10 of each of these guys. There's actually specific numbers for each that I'm not going to make you know either. But we'll take one V and one D and one J, and we'll just randomly pick them. And when we stick them together, we edit them too. So your cells will literally just remove or add random amino acids, nucleotides resulting in amino acids to really do some variability. And so there's gene editing going on for each of your lymphocytes there to aid in increasing our ability to interact with diversity. So I have a little short video on this. that will summarize that. Hopefully it will play and not be janky like it was before.
People are full of antibodies, a lot of antibodies. These proteins are a vital part of the immune system, but just how the body makes such a variety of them mystified scientists for decades. The human immune system has to fight off an endless variety of bacteria, viruses, and parasites, and for each invader, it needs a specific antibody. Luckily, it can generate billions of them, each slightly different, ready for any disease. But these antibodies, made from 2 light chains and 2 heavy chains, are proteins, and billions of different proteins should need billions of genes to make them. Humans have around 20,000 genes. The solution to this problem was a Nobel Prize-winning discovery. Antibodies are made by B cells. Like every other cell, B cells start off with the same 20,000 genes, but they also have an enzyme called RAG. RAG is a DNA shuffler. It targets the genes that make the antibody's binding site. There are 3 kinds of gene segments here, called V, D, and J. For the heavy chain of the antibody, humans usually have around 130 different V segments, 27 D segments, and 6 J segments. The RAG enzyme binds to one of the J segments at random, and then to one of the D segments, chopping off the spare DNA between them. Other enzymes then join the ends together. The same thing happens with a random V segment. So you end up with a G composed of 1 V, 1 D, and 1 J joined together. This provides over 20,000 possible VDJ combinations for the heavy chain. The light chain can have another 400 possible gene combinations. Combining heavy and light chains gives approximately 8 million different possible antibodies, each with a different binding site. And that's not all. During the joining, extra DNA is added or removed at the junctions of the segments, which takes our antibody diversity from the millions into the billions. This is how our immune system can respond to diseases it's never even seen before. In fact, there's so much diversity that one of our antibodies could potentially bind to almost any target in the whole universe.
It's pretty cool stuff that your cells have developed to do that. We see this in all sorts of mammals that have similar immune systems to ours. So it's not just going to apply to humans, but that we have this similar sort of gene reassortment going on in all sorts of animals, which I think is just a fascinating thing that our body has developed that in order to have all this sort of variety there. So once a B or a T cell that is assigned to see that specific antigen sees it, we're going to clone it out. And that's our proliferation step. So we're literally just going to make copies. So if you are the only B cell that sees red top marker and you see red top marker, then that B cell will just multiply out all the red and to make many red top marker seeing B cells. You only have one to start off with. Well, once that one cell sees red top marker, it will proliferate out into many, like millions of clones of that one B cell that sees red top marker. All right, and so we'll have all of the clones that result from that, and each from that whole slew of those clones, some of them will be assigned to be memory cells, some of them will be assigned to be regulation cells, and some of them will be effector cells. And all of that is going to be triggered by our activation by seeing that antigen and getting the other signals from our body saying, yes, you've seen this antigen and you have the go ahead to respond, right? If we're reacting to, let's see, you finally see your very own red top marker antigen, whatever your marker is, you've seen your antigen and you're ready to proliferate. What happens if that antigen that you saw that you've been waiting to see your whole existence was a self marker? Well, your body doesn't want to, tell you to go ahead and attack that. And so we have signals that are in place that will stimulate you when you're supposed to respond. And if you don't get the stimuli telling you to respond, you just don't. And then you not only do you not respond, but you'll die because you tried to respond to something without getting the go ahead. And so that's how we're going to eliminate those self-responding guys. Okay, so that whole process of first eliminating, hopefully early on in your development, your body went through this process of eliminating all of those possible guys that you made with all your gene reassortments, eliminating the ones that will react to self. So that part is called the clonal deletion process, and it leads to immune tolerance. So tolerating self is what we're talking about there. So that happens first. So eliminating, that's the other side. Eliminate anti-self happens first. Again, that's called clonal deletion. And what results is immune tolerance to tolerate self. All right, the next step is now that you have all of these cells that can react to hopefully not self, right? You're hoping that you got rid of that during your development. You have this whole repertoire of cells, and each individual cell, there's only one for each possible antigen that you could potentially encounter in your entire lifetime. And they're all waiting to see their antigen. When you finally see red triangle antigen and you interact with red triangle antigen on your receptor, then we will only populate out the cells that see red triangle antigen. The other guys will still be waiting to see their match. This process altogether is called clonal selection. So first, always the first step is to proliferate. And this whole process is involved in the proliferation step. So we will proliferate out all of these cells that could respond to the red triangle here. Then after that, they will differentiate into their functions. And the three possible functions for all of our lymphocytes. The first one is going to be memory. It doesn't matter which order you put these in, but there's three of them. There's memory, there's the regulators, so that we know that we're not going to overreact to things, or we can make sure that we are reacting appropriately to things. And then the third one is effector, E-F-F-E-C-T-O-R, like when you have an effect on something. So those are effectors. The effectors are the ones that are actually doing the work. So these are our working cells. They're doing the job. All right, we talked about immune tolerance. All of your B cells are going to be going through that clonal deletion process of eliminating self cells and maturing in the bone marrow. And they're going to wait until they're ready to see their antigen. We've been properly selected out and we're ready to go and see the world. All that maturation process happens in the bone marrow. After that, they're going to wait in the lymphatic tissues to see their antigen. For the T cells, they will start out in the bone marrow, then move to the thymus to finish their maturation. So all of their clonal deletion and prepping and everything like that is all going to happen in the thymus. And once they are naive and ready to go out into the world and wait to see their antigen, they can circulate around in your circulatory system or hang out in the lymphatic tissues. They can kind of do both, and they'll just kind of migrate around randomly in the hopes that they're going to see their actual antigen. So the T cells, it's actually extremely essential that the T cells be getting out there and doing their job. And now I'm going to walk us through what the T cells do and why and how this whole process works. I'm going to do this in the whiteboard thing. Hold on. I have this up now. Hold on. Trying to pull up the whiteboard. I feel crazy because as soon as I look at this and I'm like, I swear to God, it usually says whiteboard up here. More breakout rooms. Well, I guess I'll just pull up a blank slide then because I can't seem to see where the whiteboard thing is. Share. I've done this a million times and you'd think that I would know. Maybe it's over here. Top toolbar. Nah. Okay. It's been too long. Every semester I go through this and then I have to relearn where the freaking whiteboard is. All right, let's just open up a new slide. And where I want to do this, T helper right here. And new slide. Just want a blank slide. Like, I don't know why this has to be a, there we go. All right. cool. So I'll just draw it on here. All right, so basically, we know, we've already talked about previously how you can have outside antigens, right? They're just hanging out. They got into your body somehow. No matter if you have an inside infection or an outside infection, everybody also has outside. Everybody has outside to some extent. But if the infection is focused on the inside, then there are extra steps that we'll want to take. But everything also has the outside component. So we're going to start with our explanation from the outside. So when you have an introduction of a pathogen into your body, we'll have phagocytes that come up there and eat it up. Usually your first responders that are as your phagocytes to come eat things up are going to be your neutrophils, remember. So they're going to come in and eat things up, but they're just going to poop out the waste. They don't do anything else with it. So they'll eat things up, and then our macrophages will come in and eat up the neutrophils that did the work, as well as all the waste that they left behind. So here comes our antigen presenting cell. In this case, this is going to be a macrophage. So I'm going to abbreviate it like this. Okay, this is our macrophage. And it's going to eat up stuff that was left in that area. It picked it up from outside of its own self. So where is it going to present it on? What kind of MHC? MHC2, right, exactly. So MHC2. So just like we drew before, it's going to present its antigen on MHC2 because it came from outside. Now, just like we learned before, we have our T cell receptor, which I'll just put TR here. That's our T cell receptor that specifically recognizes that antigen. All right, so our T cells, and this one, we're going to even be more specific about because we have multiple kinds of T cells. I'll tell you how to tell the difference here in just a minute. This is going to be a T helper cell. So with our T cells, which I'll write out over to the side here, we have two different kinds of T cells. Ones that have a marker called CD4. This is just a marker that helps us identify the difference between these guys. It has a function as well, which I'll get into in a second. But CD4 marker or CD8 marker. 4, to me, if I draw a 4, this looks a lot like a letter H without one of the legs to me. These actually are our T helper cells. All right. So you're either a T helper or you are a cytotoxic T cell. Those are our two types of T cells. To me, 8 has round shapes to it like a C does. And that's how I remember that one too. Okay, so we've kind of figured out CD8, which I might just call them CD8 positive because they're called CD8 positive because they're positive for the CD8 marker, or CD4 positive T cells. Now, do you guys remember how when we talked about HIV, I told you that they infect T cells? HIV infects CD4 positive T cells. This is going to be extremely important for us understanding why HIV causes disease here in a minute. All right, so our T helper cell is here to recognize our antigen back over here in our example. All right, so what marker does our T helper cell have on it? CD4. As it turns out, CD4 is not just a marker. It helps recognize MHC2. CD8 is a marker that, which one do you think it's going to help recognize? Recognizes MHC1. All right, so we've got this idea going on here. Always, this is how this is going to work, okay? But right now, we're first encountering our antigen from outside, and that's going to be presented on MHC2. So the marker that's going to interact with MHC2 is CD4, and that means that all of your outside stuff is going to be first recognized by your T helper cells. Your T helper cells are your captains in all of this. Nobody, not your CD8 cells and not your B cells, none of your adaptive immune response can do anything without the signal from the captain. So now when I'm telling you that, you can't make antibody and you can't have your cytotoxic T cell response, which I'll tell you about what that does in a minute. You can't do any of that without the captain giving you the go-ahead. That means you can't have antibody and you can't have a cytotoxic response unless your captain is there to tell you what to do. And if you have HIV, your captains are all infected. You don't have any captains to tell you you can fight disease. That's why HIV leads to immunodeficiency, not just because it's killing off your T cells that do immune work, but because it's killing off your captains. Now your B cells, which are fine, they're not infected, but now your B cells can't even do their job because they're not getting the signal from the captain telling them that they can. Suddenly, T cell deficiencies led either by HIV infection or by genetic abnormalities, suddenly T cell deficiencies are a significantly worse aspect to have than just a B cell or immunoglobulin deficiency. Because if you have a B cell problem, your B cells can't work, but at least your T cells can still do a thing. If your T cells don't work, your B cells can't do a thing either. This is double bad if you have a T cell problem. Okay, so this is our captain. So we're activating our captain, and this captain will basically signal everybody that they can do anything. So now we have interacted with our antigen that we saw, and our CD4 molecule has come up as well and done its job by verifying that, yes, this is coming from Lauren's MHC2 molecule. It's coming from outside, and I'm recognizing my specific antigen. We're recognizing that it's coming from Lauren as well. All of that is giving a signal saying Lauren's cell found something from outside, and it matches with my antigen. Everything is in order here for me to start signaling my other cells that if they see this same antigen, that they can attack too. All of this has to happen first, which is why we're starting with it first. Okay, now we have our T helper cell activated. Well, this guy, our T helper cell, differentiates into two. Actually, let me go ahead and do this. The T helper cell can differentiate into two types, TH1. There's other types, but I'm just going to have you guys learn two different types, okay? TH1 and TH2. So T helper is the naive guy. He's just the T helper cell that can see that antigen. Once he sees his antigen and gets that signal saying that MHC2 is presenting it, now I can, remember what we said, we proliferate and then we differentiate. We proliferate out the TH, and then the THs differentiate into its memory, its regulatory, and its effector. This TH cell has two effector cells. We'll also get memory and regulator. I'll put them up here so that we don't forget that they also happen. All right. So these are our effector cells. We have two different ones for this guy. It's the only one that has two different effector cells. The TH1 response is going to regulate inside infections. The TH2 response is going to regulate outside infections. So we have to get the initial response from outside. Then, no matter what, we saw outside first. No matter what, though, our T helper response, once we're activated, we make both so we can be ready for both. Because we don't know yet if there's inside infection or not. Let's say you have an infection that is all outside of cells. So this could be a bacterial infection, like a staph infection. Those guys don't ever get inside of your cells. They get in your tissues, but not into your cells. So that's all outside. So TH1 cells will still be made from this, but they don't really have any role or function. They are not really going to be regulating anything with the inside stuff, but we made them just in case. But if you have a viral infection, who do you think is going to be the busiest? TH1. So this is now where we're going to get that differentiation going on. Okay, so that is how everything starts. Hopefully that'll write. Where did it go? Did it add it at the end? Is that what it did? I'm gonna lose it. I'm gonna keep it, I guess, because it's like I just drew all that stuff and now I'm like, where did it go? Here it is. Okay. God. Okay. Alrighty. So so that's where we're gonna start everything. I don't know why it went back like that. I didn't push any button. Whatever. So we've got our captains, our TH1 captains and our TH2 captains. So everything's going to work like that. And I'll draw out another thing to go along with that once we get to the big picture thing. But first, I want to start with what's going to be going on for either inside or outside infections separate from our captains. So for the second part, inside, let's go look at our inside stuff now. You have an infected cell. This is our infected cell. Here's our whole cell. Here's the viruses that are inside of it. So this is an infected self cell, and it's got stuff inside. Which MHC is it going to be using to hold out the antigen? MHC1, right? So we've got our antigen on MHC1. All right. We've got our T cell receptor. And this is on, what kind of T cell is going to be able to interact with MHC1? The CD8 positive cytotoxic guys. So this is coming back up here. Okay. So this is a T cell. This is cytotoxic. And it has a marker too. It's also CD, but this one's CD8. And this marker on the CD8 positive cytotoxic T cell. Now our receptor recognizes the antigen. Our CD8 marker recognizes that Lauren's cell is holding up an antigen from inside. So we're getting a signal. My antigen that I see is here. Lauren's cell is holding it up on a marker saying that it was inside of her cells. And we're getting the signal that all that is going on. This cytotoxic T cell can't do shit until it's TH1, this guy, comes and says, this situation is okay, and you have the go ahead. We clear? All right, so that's what happens with that stuff. Then, once we get this activation signal from the TH1 saying we can do stuff, then the cytotoxic T cell will make things called granzymes and perforins. What do you think perforins do? They perforate. Okay, so they make holes. And so the granzymes and the perforins here are going to come over here to this cell, poke holes in the infected self cell, and then the granzymes are going to digest everything inside of that self cell and kill it. And I'm saying that all this happens is that at some point in your body, an outside thing was found. The outside thing was found and triggered your T helper cell. The T helper cell activated and made a TH1 cell. The TH1 cell found your cytotoxic T cell that saw its antigen in the infected self cell asking for help and said, go ahead and kill it. And then it releases granzymes and perforins and kills the infected self cell. But all of this happens like that. And these cytotoxic T cells, once they've been activated by our T helper cells and the signals they're sending, once they've been activated once, they can go around and keep doing their jobs. But they have to have that initial go ahead from the captain. And now our cytotoxic T cells can literally just go around, have this interaction, and have the verification on their own now, and just release granzymes and perforins. And when you see videos of this happening in real time, it's like your T cell comes up to the infected cell. The T cell is much smaller than the infected cell, by the way. It's this giant, massive, almost building. And this T cell comes up to it, makes this interaction, and bam, that thing just dies. And it just, and it's gone. Okay. So that's crazy that every time you have a viral infection, this is going on in your body. And now you wonder why you feel like crap when you get sick. It's these T cells. But that's also why we have to have regulation. That's why we have to have the captain there telling it to go ahead. That's why we have to be sure we're having that specific antigen interaction. And there are plenty of other signals going on to help mediate this response as well, because you don't want to just go around killing all of your cells in your whole body without some sort of regulation going on. And this can go awry. If your antigen that you see is something that isn't actually harmful, but your body has been trained to see it as harmful. This is where we get allergies and things like that. Anybody in here allergic to poison ivy? Ever have a reaction to poison ivy? Yeah, come on now. Almost all of you probably have at some point. If you haven't interacted with it, you're going to tell me that you haven't had a poison ivy rash. If you get a rash from poison ivy, you're allergic to poison ivy, period. Do you know there's people that don't get rashes? That people that do get reactions to it? No. No, yeah. My ex-husband could roll around in some damn poison ivy and never have a rash. So you don't just automatically get rashes from poison ivy. It's an allergy. So your T cells, what happens is we have allergies that are mediated by antibody, or we have antibodies against dander or something like that. And you'll get hives and you'll get itchy and whatever. But when you have a T cell that recognizes something as foreign and it attacks, it's killing your cells thinking that you're infected inside with something. Poison ivy does that. So this is what we call contact dermatitis. So there's eczema, which is like when you get hives and itchiness from that. You can have dry skin and the itchiness and the redness. But contact dermatitis, you have the weeping and the actual damage of the cells as a response to the thing. So contact dermatitis is driven by your cytotoxic T cells. Your cells think they're infected inside with something, but it's really just the oil from the poison ivy that's making the cells think that. And so your body is what's causing the damage. The oil on poison ivy doesn't cause damage. It's actually completely harmless. Your body is attacking it, and that's what the harm is from the poison ivy oil. And that's what's actually going on. And so some people don't have that because their antigens didn't get a response with that when their bodies were reassorting their genes. It's a pretty rare thing to have. They're not common. Most people have a reaction to it. But the plant started developing this oil. It didn't think in the plant, this is a toxin that I'm making, but it happened to have this oil that it made, and things stopped eating it as often. And so it was a benefit for it to have that. As it turns out, most animals will have that sort of a response to it, where their T cells think that they need to be killing self cells because there's an infection going on. We'll talk about a little bit more about that and how it works when we get to chapter 17, but that's issue when your immune system goes wrong. Yeah, similar to that, right? When your immune system isn't attacking actual pathogens, but is attacking something else, right? So this is how our inside stuff will be working. So let's touch really quick on our outside stuff. If you have an outside infection, we've already said that our outside infection will lead to our activation of our T helper cells. But all that your T helper cells do is regulate. They're not fighting anything themselves. They're just making sure that the fight is going on the way that it's supposed to. So otherwise, when you see outside stuff, you have crap floating around in your lymph and in the lymph node, you will have a B cell. And on that B cell, we have our B cell receptor. And you guys know this guy well at this point. It's an immunoglobulin. As it turns out, the receptor on B cells that's bound to the surface is immunoglobulin D. It's a version dictated by that constant region. The constant region on this makes it the IgD, and the IgD makes it the receptor. It's on the surface of our B cells. So this is basically the B cells waiting in your lymph node, just waiting to see that antigen. And the way that it's going to check that that antigen is the antigen it needs to see is by using its receptor, IgD. And so it binds to the antigen, takes all of that inside of itself, and presents it on MHC2, because it came from outside, right? And then we can get activation from the T cell receptor, and that would be the CD4 on our T helper cell, right? And then it would do its whole thing that we did up here. So that's the way that B cells can act as antigen presenting cells. You remember how I told you they're not phagocytes, but they're still antigen presenting cells. That's by acting with that B cell receptor, bringing it in, saying my B cell receptor found this outside, put it on the MHC2, and then does the exact same thing as what we saw up here in the start. So that's how we can start one of these reactions. What if the reaction is already started, though? What if we already have a Th2 guy out there? Then we don't need that activation step to happen. Then I'm trying, man. Okay. Then the similar thing will still happen where they will take it in on their inside and we'll get our signal from our T helper, but it won't just be a naive T helper, it'll be a Th2. And so when Th2 comes along and we have MHC2, we'll just put whatever, MHC2, fine. And we have our T cell receptor that interacts with it. We have CD4. But now Th2 mediates this response. It basically can activate the B cell. And what is the B cell going to do when it activates? It's going to make antibodies. And so B cells, whenever they activate, they're also going to turn into memory cells, regulator cells, and plasma. Plasma cells are B cell effector cells, which have their own name. So the effector cells for the T helper cells, they're Th1 or Th2. The effector cells for the cytotoxic T cells are just cytotoxic T cells. The B cell effector cells are called something else. They're called plasma cells. And plasma cells are, as our effector cells, they're the ones that make antibody. See, when I say that this is complicated, just trust me when I tell you that it's actually a lot worse than this. And I'm trying to give you the basics here. All right, so now our plasma cells are the ones that are going to crank out antibody. We caught up on this. Are we starting to see how these things are going to regulate each other? We're getting the idea of it.
What is that arrow when you're at the Th2 and it goes straight to the B cell? What is that?
That's activating the B cell.
Okay.
Because otherwise the B cell interacting, you see how our B cell, our receptor on our B cell sees the antibody, it sees the antigen, right? The receptor, the IgD, sees the antigen. It can't make antibody just automatically. It has to get the signal from the captain. And so that's what that is demonstrating. Activation from the captain. B cells cannot activate and make antibody unless the Th2 cells tell them they can. Cytotoxic cells can't go around killing infected cells unless the Th1 cells tell them that they can. So those Th1 and Th2 are going to mediate how much outside and how much inside response you need. If you don't have an inside infection, your cytotoxic T cells aren't going to be engaging with CD8 and their antigen, and you won't need Th1 to mediate that. But if you have an outside infection, you still need an antibody. And so your Th2 cells are going to mediate that antibody response and make sure we're making antibody, but only when we need to be making antibody, right? So we don't need Th2 activating B cells that are trying to respond to self antigens, for example, right? And it's when that system goes wrong that you see autoimmunity. When you start getting signals from the inflammatory system or from the Th2 cells getting signals to tell them to go do this work, because we have other infections going on, then anti-self cells will get cross-reacted. Otherwise, normal situation, anti-self cells won't be getting the signals. But if you've got some other infection going on, you might accidentally activate the anti-self cells at the same time. And that actually happens quite a lot. We actually believe that a lot of our autoimmune diseases are triggered because you have another infection and we accidentally triggered anti-self during that response. We don't know how to control that really, unfortunately. Okay. Plasma is just the name of the antibody producing effector B cells. So effector B cells that make antibody are called plasma cells. Yes, Shy. Yeah. Mhm. No, B cells do not act in infected self cells.
Okay.
That doesn't help you, right? So if you have stuff that is inside of the cells, antibody is protein that acts outside. The antibody can't get into your cells. So antibody acts outside. on outside things. But if you have infected cells that are infected inside, then the only thing we can do for infected cells with stuff inside of them is kill them. That's the only thing we can do for the infected cells. But if you have a viral infection, do you only have things inside of the cells? No, right? Because when you have a viral infection, you have stuff inside of the cells. We can't allow them to propagate, so we kill the infected cells, but there might already be virus hanging around going to infect new cells in the outside. There, in the outside, antibody can block those guys. So when you have an inside infection, you need both, right? You have to have both going on. And so it's important to remember, you don't always have one or the other. It's almost always going to be both going on to some degree, right? Does that help?
Yeah.
Okay. So let's move on, because I know it's a lot all at once. We already did all this stuff. And I'm going to skip over the slides that talk about the stuff that we just covered, because words, and if you want more clarification, please feel free to go back and read those slides. But I tried to walk us through the actual events so that we could keep it in terms of how it's actually happening in the body. There is a video here that explains the T help response a little bit better if you would like to go watch that. And then there are some figures here that talk a little bit more about T cell activation. We already mentioned cytotoxic T cells produce perforins and granzymes, perforating our target host cell, like your own self cells, to let the granzymes digest the stuff that's in them. This will lead to a process called apoptosis, which is cell death. And so whenever your cells know that they are at their end, that there's no like fighting back, then they'll go through something called apoptosis. It is essentially a cell suicide. When it knows that it can't come back from a thing, you'll get enough signals knowing that, and that's what will happen due to perforin and granzyme. These cytotoxic T cells don't just attack virally infected cells, they also attack cancer cells and cells from other humans and animals. So these cytotoxic T cells are the ones that are going around and recognizing that your kidney is not my kidney and attacking it. So we're literally just killing those kidney cells from somebody else, just because their MHC1 doesn't look like my MHC1. And so it recognizes that and just initiates the whole response. So that can be lethal. And so we sit here and we say, so we have a mechanism in place in our body right now that actually fights off cancer. Like that can't be possible because people still get cancer. Let me tell you, go look at somebody that has HIV. Somebody who has HIV that cannot have a T cell or a B cell response. They get a ridiculous amount of increased frequency of cancers that you will never see in otherwise healthy people. This includes things like Burkitt's lymphoma or Kaposi's sarcoma. These are cancers that otherwise healthy people would have. You, me, and everybody in here would have them if we didn't have our CD8 T cells destroying them. And their CD8 T cell response doesn't work because their captains can't tell it to work. And so they get these weird cancers that we would have otherwise, but they can't fight them off. And so when we talk about Burkitt's lymphoma, this is a lymphoma cancer that is caused by having had mono. And 90% of adults will have had mono already. And so you've probably had mono, whether you knew it or not. I've never had symptoms of it, but I have antibodies in my blood for it. And so I will have had mono. And it's one of those things like chickenpox and herpes. It's a herpes virus that when it gets in your body, you have it forever. It hides out forever. And those cells are constantly going to have that mono DNA in them. And your T cells are constantly fighting off Burkitt's lymphoma from happening from you having had mono when you were a kid or something. But it's happening right now in my body. And it's pretty impressive to think that some of these cancers can be controlled that way. Now, unfortunately, not every cancer calls out for help with the MHC like that. Yes. Mm-hmm. So this is a thing too that I want to mention. That's not entirely uncommon. I have usually probably every semester somebody will bring up that they have a loved one that has chronic mono. It's like chickenpox. And you know how some people, when they have chickenpox, they'll get shingles, like even at very young ages. And it'll, yeah, and it'll just keep coming out and keep coming out. Well, people get mono like that too. Once you have mono, you have it forever. And your immune system is keeping it under control right now. My immune system is keeping it under control. But you said your sister, right? Her sister her immune system isn't able to keep it in control. So it keeps coming out. Her immune system just isn't as effective at fighting mono as ours is. Now, the likelihood for it becoming Burkitt's lymphoma would entirely be based on how her cells will respond to the cancer aspect. So it could still be that her immune system is strong enough to keep the cancer aspect of it aside, but it just can't keep the viral replication of it aside. So it'll be a different kind of response for that. It's a really good question. Yeah, and it gets really complicated, as I'm sure you guys can tell. So we talked about the B cells and how they make antibody. Now let's get into antibody and what it does, okay? Okay, so these are our functions of antibody. There's 6 major functions. We can coat the surface of bacteria, like coating the surface of something with flies. When we had in the previous chapter, there was a little video on there on complement, and it's like, oh, complement is basically like coating something with flies. Antibody can do the same thing. If you were coated with flies right now, you would have a hard time functioning. And so will the bacteria. If we coat it in antibody, it has a hard time engaging with the cells. It has a hard time engaging with other bacteria, and it has a hard time getting around in the body. So we can just coat things and just prevent it from being able to function. All right, we have opsonization. We introduced this term in the previous chapter as well. So opsonization is basically the antibodies are sticking to the outside of the capsule or the outside of the bacteria or whatever it is, and it gives an easier handle for our phagocytes to grab onto. So opsonization is helping phagocytosis by helping them grab onto the pathogen. We have coating. We can help it grab onto the pathogen. It's a term called opsonization. We have neutralization, where our antibody literally binds the spike or whatever else that pathogen uses to get inside of cells. So this is a specific interaction. We're not just covering something and blocking it from binding. We're literally binding to the key so it can't get into the lock. Okay, we have agglutination. This is causing the guys to stick together. If I coat everybody in here with a super, super sticky, tacky glue and then push you all into a big old clump, you're going to have a hard time getting to your next classes, getting into your car, going home, anything. And that's the same thing with these guys. We're going to cause them to just stick